for molecular genetics at the University of Berlin. He's German, as you will no doubt figure out when he starts speaking. He then 7 years ago moved to the United States, and he did a postdoctoral stint at University of California, Berkeley. And he was also a fellow at the Berkeley Institute for Data Science. He has now moved, still in California, but to San Diego, and he's now assistant professor at UCSD. John comes initially from a purely modeling background, but he saw that really it is the bridging between modeling and biology that is the future. And so he has branched out into actual experimental research, as well as developing new modeling technologies and approaches. And so he's going to give us pretty much what we are all striving for, the beautiful combination of both experimental biology and cutting edge modeling and computational approaches. Jonathan.
Oh yeah, lights down would be fantastic, actually. We have lots of, yes. Yeah, just a little bit. Yeah, I realize we just come from lunch, but we have lots of microscope movies, so it's actually good to dim the lights a little bit. Okay, good. Thank you so much for that introduction. Yes, it's true. We are really trying in the lab to bridge modeling and experiments. And I'm really excited today because we brought something that we just published yesterday. And that's where this video is from. So that's going to be the second part of the talk. The first part of the talk is essentially how we really approach whole cell modeling from the modeling and experiment perspective. And so my first slide is essentially about this. So what I have on the left-hand side, that's essentially what I call bottom-up simulation. So typically you start with system parameters and then go into some simulation engine. For us, that's essentially this ready reaction diffusion dynamics particle simulation engine. And then that goes into different algorithms to essentially create something that is comparable to the other side. And the other side is, in our case, the experiment. So that's the imaging in our case. We do very advanced microscopy. So that's the AOLLSM, that's for adaptive optics lattice light sheet microscopy. That's arguably the most advanced microscopy technology that you can do. And so we are trying to essentially combine these two, address a biological question by combining these two, and use deep learning as the bridge for the communication between. And so what I brought today are two examples where we applied this framework to biological questions. The first one is about clathrin-mediated endocytosis, and the second one is going to be about mitochondria and organelle dynamics. So this is all the published stuff, so I'm going to be very quick. This is all the unpublished, as in, like we put this on bioRxiv yesterday, so we're really excited. Yeah, you can check it out. So yeah, this is going to be the first presentation of this work, actually. And it was done by Zach. Zach is in the audience. He's going to give a software tutorial about how you can track organelles and mitochondria tomorrow. So that's going to be very exciting. So just show up and we can all do this together. Okay, so let's start with CME. CME is short for clathrin-mediated endocytosis. And so clathrin-mediated endocytosis, that's essentially how the cell takes metabolites, proteins, hormones, and receptors, and also incidentally COVID-19 particles into the cell. And the main two proteins to really keep in mind here are clathrin, that's essentially the cargo container forming protein. And then you have dynamin, that is essentially the molecular scissors protein that then cuts the vesicle off of the plasma membrane once the vesicle is fully formed. And so then if you're a cell biologist, what you really care about is the dynamics of how this process happens, when the individual protein is recruited, how is this related to different metabolites, to different proteins, different conditions, different diseases. That's essentially one big field in cell biology, trafficking and clathrin endocytosis. And so the way we approach this from a modeling perspective, this is a busy slide because we did a lot of work in this already. So this is the READY slide, essentially. So READY stands for reaction diffusion dynamics. And it's essentially on a mesoscale level for doing cell simulations, okay? So typically, like what we heard in the first talk today, that's like you really simulate the full cell, but there's not much spatial information. This one is going like much higher level of detail in terms of space, but then like it's not like the whole cell, but it's like small parts of the cell essentially. And so typically one particle is one protein, and so then essentially it's an integrator for overdamped Langevin dynamics. So people who are familiar with molecular dynamic simulations, this is essentially a diffusion particle dynamic simulation. So I wrote that in my PhD. So there was like a little bit of work, and then we applied that to clathrin endocytosis. And so what you can do here is essentially do diffusion simulations of how these dynamine particles are aggregating around these necks and how they then diffuse and form these scission reactions. We can also apply it to synaptic vesicle release situations. So this is the top one, the clathrin one. This is really one particle, one protein. The synaptic vesicle release, that's essentially one protein subunit, one particle. So you see essentially these synapsin and other molecules that have like these little flexible loops. So this goes more like into the like little coarse-grained MD. And then here, this is with Blair Lyons from the Allen Institute, because it's super exciting. And there's going to be more from Matt and Iran, I think, tomorrow about this, like how you can integrate READY with the Allen Institute for Cell Science visualization tools to really make to do amazing work. So I'm really looking forward to that too. So that's essentially how like this is all published. I'm not going to go further into details, but this is how we approach this clathrin endocytosis from a modeling perspective. Okay. So then let's go to the imaging perspective, and that's I'm super excited about this because I really think we're going to see a revolution in the way we do cell biology. Before, or like essentially the last 20, 30 years, we essentially did 2D cell culture. We then did 2D plus minus time imaging of those cells in culture. And then you would essentially do a semi-manual data analysis with like fantastic tools like Fiji or ImageJ, but you would still like use your mouse to essentially like outline your owl eyes and your interesting phenotypes. That is going to change very rapidly. In fact, it's changing right now. What we are doing in the lab, we essentially take human stem cells, we CRISPR genome edit them, and then we grow three-dimensional organoids from those stem cells. Currently, we have intestinal organoids and brain organoids. But then you can, with these, with the development of adaptive optics status light sheet microscopy, you can go ahead and image these three-dimensional organoid tissues live. So you can do cell biology in living tissue. And now you really create 3 terabytes per hour. And so you're not gonna like take your mouse and like you have this massive data set. It's like 3-dimensional. Like it's actually like we experiment with the virtual reality a little bit. To like even, but you're not gonna click on anything there, right? So you really need like new tools, big data and machine learning tools to do the quantitative analysis, and that's what we're doing. So our lab is really focused on this part. And so what makes this possible is this technology. It's called adaptive optics lattice light sheet microscopy. We can go into all kinds of details, but what the takeaway is, it's 100 times faster than a confocal microscope. It's 100 times less photobleaching than a confocal microscope. And then it has the adaptive optics. So what you can see essentially, so this is a little bit of like evolution of microscopy. What you see is you cannot really buy the microscope itself. Okay, what you buy is like a couple hundred to low thousands of parts, and then you like buy your optical table, and you also like construct an optical room actually to like put it in there. Exactly. And so yes. We have actually an interesting garage story. Ask me later about our garage story. this is our garage story, in fact. So we for all kinds of different reasons, we had to build the prototype of the microscope in an office. So this is actually us like rigging the office, because you cannot really drive six lasers, like high-powered lasers, in an office. So we had to like draw cables from the ceiling to the whatever. So yeah, but yes, so we have this. We built it. We have it online. Really proud. So this is Hiroyuki Hakusaki. He's like really our fantastic microscope engineer. It's ready. And so what it does is, so the adaptive optics, what it really helps you to do, like when you look at your hand is not really see-through, okay? So if light goes into tissue, it gets aberrated. And so what that means, if you actually want to look at like really small details of cell biology, like clathrin endocytosis vesicles, then you just, in an organoid context, you're just gonna see this blurry mess, okay? With the adaptive optics, and I have this little earphones over there, that's essentially, the analogy is a little bit like you put these on the airplane to essentially shut down all the noise. So they essentially, we do the same thing with photons. We can essentially, get rid of the optical aberrations. Yeah, so the bottom line is you get beautiful data sets like this. Okay. So this is now on the top left, I have an intestinal epithelial organoid. So we are looking only at the top part. So these are like maybe 30 cells that we're looking at. You can kind of get a hint of the outline of where these cells are, because the endocytosis is happening predominantly on the plasma membrane. So yeah, and each of these individual dots is like one clathrin-mediated endocytosis event. So it's beautiful, like you see, like we used to do these imaging studies in turf microscopy. So turf microscopy means, you literally only see what is in your turf field. So that means if something gets, say, like budded off and goes into the cytoplasm, you lose it. You have no way to access the lateral sides of the cells or the apical side of the cell. So with this, we have like access to absolutely everything. So it's wonderful. Problem is, we are dealing with 3 terabytes of data per hour. So that's we became very quickly the biggest single data producer on campus. Like it used to be, yeah, it's a problem. And so when you have this, the problem is then, the classic microscopy, that's maybe a gigabyte of data per hour, and then there are tons of visualization tools, tons of quantitation tools, and you can do this manually. For us, we really have to go into high performance computing, because the data is so big. We have to really go into machine learning, because that really gives us the power to actually pull the quantitative information out. And we have to develop these, because that is not really around. We are the first people to have access to this microscopy technology, so we are the first people to create those data sets. So we have to develop the technology to process it. And so this is one of the first tools that we developed. So this is called PyLattice, for Python-based lattice light sheet image analysis. So this focuses on individual puncta, so these individual vesicle tracking. So this is all on GitHub. It's all published. You can check that out. What it does is it detects all of these individual events in the 3D data set, and then tracks them over time. Okay, so this is essentially like tracking of these individual clathrin-mediated endocytosis events and segmenting these. And then, you can essentially do your cell biology now on these big data sets. So that means, for example, you can look at one individual event, and you look at the diffusion that is very confined when the vesicle is still on the plasma membrane. And then when it's cut off by the dynamin, essentially like it's much faster now that it's freely diffusing in the cytoplasm. That's one. But then you can do this for like all of those events, and then look at like individual clathrin-mediated endocytosis cohorts of like faster and slower endocytosis events. And so yeah, that's all published. But that's essentially the direction this is going. So then that is the imaging part of what we're doing. Okay. So now let's look at the deep learning. So where we use deep learning and machine learning for a couple different use cases. The use case that we use it often these days is actually to create ground truth training data for segmentation. Okay, so typically what you would do, if this were like a two-dimensional data set, what you would do is you would essentially have your two-dimensional data set. You would give it to a couple of students to say, look, please trace the outline and create ground truth data so that I can train my deep learning TensorFlow model or PyTorch model. And then I have essentially my automated segmentation tool. Okay. In a 3D data set or 4D data set situation, that is much, much, much harder. I'm actually going to like give a student like a 4D data set, and then the person has to like essentially go through each individual slice, and then like it's actually becoming very, very impractical very, very quickly. Okay. So then what you do is you essentially use simulations for this. Okay. So we essentially do particle simulations. You convolve the particle simulation data with the point spread function of your microscope. And then you essentially have created something that's similar to an artificial microscope data set. And you then feed that artificial microscope data set as a training data into your machine learning tool. Because you essentially have the ground truth data because you have simulated all the particles, you know where they're going in the next time step. And so that's essentially how we're doing this. It's obviously like three-dimensional convolutional neural networks. And so this is another application where when you use actually deep learning versus the classical tools, what you do is you can essentially get much deeper into the noise floor to really retrieve your information. So for classical tools, you really have to have high signal to noise ratio to segment your data set. If you train a very good model, you can essentially really be a little bit more benign with the laser power to your sample and still get good data out of this. Incidentally, it's also faster because once you have trained the model, you have essentially upfront invested all the time to train your machine learning model. To pull inferences from that is actually much quicker than if you would actually go the classical route and do the tracking yourself. So that's how we're doing this. So yeah, that is essentially the bottom-up simulations, creating like essentially artificial microscope data sets, for example. The top-down imaging, you create system parameters, like by essentially segmenting, tracking, and getting particle coordinates. You can really get the system parameters that you can then feed back into your simulations and essentially have this nice feedback loop to like correct yourself and see if like your models make sense or if your biology is actually if that makes sense to really learn something about the cell biology. Okay, cool. So then now the fun part. So this is essentially the data flow outline. So scopes create a lot of data. We have kind of a handle on that. There are data pre-processing steps, and we kind of know how to deal with them. The retrieval of biological information, that is really the problem that everybody has. So the particle tracking problem, that is solved. Okay, so that like Pilot is one tool. There are other tools out there that's solved. But there are like organelle membrane tracking, nuclei tracking, and novel biology problems in those data sets. Like the microscope doesn't really stop at only looking at puncta, but we can essentially look at anything. And so that's what we are now doing. We are essentially moving down this list. Next one is organelle tracking, and today I'm going to reveal MitoTNT, which I like to best of my knowledge is the first tool that allows you to track mitochondrial networks in 3D lattice light sheet data, like 3D over time 4-dimensional data sets. And so the reason one should worry about mitochondria and organelle dynamics is this. This is not what mitochondria look like. Okay, so it's very interesting. I'm going to like for fun go on Google Images and put in mitochondria. What you're going to see is like tons of these potato-shaped things. That is not what mitochondria look like. Okay, it's just not. It's wrong. I know where this comes from. It's like from the early stages of like electron microscopy. This is closer to what mitochondria look like, but this is also not really what mitochondria look like. Okay, so this hints already. This is like a 2-dimensional microscopy image of a cell that is grown on a glass cover substrate, super flat. It hints at mitochondria actually being this network. It's not like single individual potatoes or anything. It's actually 3-dimensional networks that are, and that's actually the next part. that are very, very quickly remodeling themselves. So it's a dynamic, extremely fast network. So everything in white here is a mitochondrion. What we're actually looking at here is it's two processes. So this is essentially a, I can show you the video later on my screen where there's no lag. It's essentially, there's the dividing cell, and you see essentially how all the mitochondria are swirling around and forming and reforming this mitochondrial network. They're fusing and they're fissioning, and they're doing all of this dynamics constantly, okay? And so, you know, you have to investigate this in 4 dimensions, 3 space dimensions and time, to actually understand the mitochondrial network. And so it's perfect. It's what our technology is built for. So that's what we're going to do. This is the actual data sets that are coming from these adaptive optics lattice light sheet microscopes. So labeled here is in green the cell membrane, in blue that's mitochondria, and in cyan that's the endoplasmic reticulum. What you've just seen is essentially looking at a tissue block that has been imaged by the lattice, and then it's an artificial, essentially an engineering view of like blowing the tissue apart after the fact, and then looking at all the individual constituents of the tissue, like the cells essentially. And so then you can zoom into individual cells here, and then you know, look at the mitochondrial network dynamics in individual cells in live tissues. So that is why I really believe that this technology is going to absolutely change cell biology. You know, you're no longer looking at cells that are on a glass cover slip and essentially look like pancakes. But you look at cells embedded in their tissue, and you can investigate them. It's super exciting. So yeah, but yes, so mitochondrial network, we talked already about it. It's like very dynamic. So now the question is, well, how are we going to quantify this? You know, like if you just look at an imaging data set like this, it looks beautiful, but you cannot really do anything with this, right? Like you wanna study different drug conditions. You wanna know what is the track of like an individual mitochondrial network. How often is this like fusing, fissioning? What is the size distribution here? Because this is eventually going to give you the parameters to plug this back into simulations to build like a cell simulation out of this. Okay, so yeah. So how can we get there? And so this is now when I'm essentially going exclusively to talk about Zach's work and his co-workers. So this is a lattice light sheet data set. And this is the first 2 frames of this data set, or like a zoom in of the mitochondrial network of this data set. And blue is essentially the mitochondrial skeleton. So after you have segmented the mitochondrial network, you can skeletonize it. And that's essentially how it looks like. Red is the next time step. And so what you can see from this is a couple of things. First thing is it's very dense. Okay, so it's not like a single particle that you can just like follow around. You know, so looking at distances is actually going to be very, very tricky. What you also see is the overall topology of the network stays relatively constant. Okay, so you know, you still have like a large chunk here. That large chunk has essentially like moved a little bit, but it's still that large chunk. Okay, and it's the same is true for the overall structure of the network. The topology is more or less conserved here. And so we had the idea to use the topology as a way to devise a tracking tool of the mitochondrial network, how it moves. And so we can go into the details later, but essentially it combines distances. So current time frame, where did it move to the next time frame, and topology, like what does the overall network look like from this time step to the next time frame, into a linear assignment problem that you then essentially all combine, solve, and then you get essentially these individual mitochondria tracks. So this sounds great, you know, like it's fantastic, but the question is, okay, how are you going to know if what you build is actually accurate? And this is now where the simulations come into play again. Okay, so we used READY. This time, these individual particles are no longer like individual proteins, but these particles are now subunits of a mitochondrion. Okay, like typically we discretize the mitochondrial skeleton on a pixel basis, so 200 nanometer discretization. And so then you can almost think about these things as like a kind of like 300 nanometer, like 200 nanometer diameter balls of like discretization of the mitochondrial network. Okay, and so then you do what you do in Ready. You essentially like you plug in diffusion as your main mode of motion. You have fusion reactions, you have fission reactions for mitochondrial fusion, mitochondrial fission, and then you essentially run your simulation. And so this is actually quite close to how the actual mitochondrial network looks like. So we try to really model the distribution and the dynamics based off of like our experimental data. We can go into the details of how we can build this later. But yes, so we essentially said, okay, let's use an in silico model of the mitochondrial network and test our algorithm if it can track something like this. It can track these spaghettis, okay? And so, yeah, and so then the first thing that we realized, if we only apply distances, like distances from one frame to the next frame to track the mitochondria, it's absolutely not going to work. It's way too dense. It's way too convoluted. So it absolutely doesn't work. You know, like we lose the accuracy very, very quickly. So the x-axis here is, you can think of this essentially as frame rate of your microscopes. And the yellow area is essentially what our lattice light sheet can do. So this is like between 1 volumetric frame a second to like 0.2 volumetric frames a second. Yeah, and so yeah, distance only, you lose it like super quickly. There's no way we can track this. However, when we then go for distances plus the topology, suddenly our accuracy is amazing. So on the top left, so this is essentially our mitochondria in silico simulation. Each green arrow is a correct assignment of a mitochondria, of a track, and each red arrow, and you don't really see many, is a wrong assignment compared to the ground truth, okay? And so we are actually like extremely accurate without remodeling, okay? So if we just keep the topology constant, then distances plus topology is fantastic. However, mitochondrial networks are not really, you know, they actually do have remodeling. So yeah, so here you see a couple. So this is now the mitochondrial simulation plus remodeling. So there's fission and fusion in there. And here, you know, we are a little bit in between, but we are still like above 90% tracking accuracy in the region that we really care about in these lattice light sheet microscopes. So it's awesome. So at least like it looks super cool for the in silico simulations. Okay. So now, how does this actually look like in real data? So this is the real data again. Okay. So this is like 0-second mitochondrial skeleton, then like 3.2 seconds later. And if you track the real data, it looks fantastic. It's really like, I don't know, it's, yeah, Zach, fantastic job, seriously. It looks absolutely fantastic. So we can really confidently track the mitochondrial network, really go in there. And so we call this MitoTNT for mitochondrial temporal network tracking. And so what it does is it takes, you know, 3-dimensional time-lapse data, so 4-dimensional lattice light sheet data. You segment individual cells, and then when you look at the mitochondrial network, how it moves in these like 3-second time intervals, you then essentially segment the mitochondria, you skeletonize the mitochondria, and then you track them. And so again, blue current frame, red next frame, and black is essentially the tracking arrow of where the mitochondrial network is going from this time step to the next time. So yeah, I think it's super cool. So now, okay, why is this cool, and what can we do with this? So the first thing is, you know, we can look at diffusivity, like really pull out parameters for cell simulations. Okay, so the top one is essentially fragment motility. Like if you look at like a whole fragment of the mitochondrial network, how fast is this diffusing, or is this actually diffusive motion or no? And yes, it's a diffusive motion, and we can pull out the diffusion coefficients. Then if you actually were to simulate these as like individual small particles, what would be, like, is this a diffusive motion? Yes. And can you pull out the diffusion coefficient of those? Yes. So that's essentially for the modelers. Then this is essentially, you can really look at the interesting dynamics of these mitochondrial networks as they are like being remodeled in the cell. So, you know, there are these like one fragment might essentially rotate in the cell. There's this one that essentially does this concentric motion. And then this is just like a large fragment of the mitochondrial network that is doing like a convolution of many different modes of motion. And it's super interesting. And this is literally just the very, very beginning of us being able to understand this, because before there was not the microscopes to get the data. And then there was not the computational tools to quantitate the data. Now we have both of these. And so now we need a couple of cell biologists to really say like, gosh, this is awesome. Get Joe on the phone and let's work together on this and really do some more cell biology. Yeah. It's on bioRxiv since yesterday. And then so this is just the statics. This is essentially mitochondrial dynamics. So this is like fission and fusion. And so since we have like a super high resolution grasp of what's happening in the mitochondrial network, we can essentially look at node level fission and fusion of the mitochondrial network. And so we call these skeleton nodes. And so what we see is if we control for everything, the nodes that are undergoing fusion and fission, they actually have a different motility than the other nodes and just random nodes. And that's actually quite interesting, because I think that hints at like more interesting cell biology of like the molecular machinery that is actually like, you know, this doesn't happen in thin air. You know, there's the endoplasmic reticulum involved. There are like all kinds of like different molecules that have to come and cut the mitochondria. So this is essentially a hint of those effects I hypothesized that we are seeing. And then, so this is just a hint of the direction in which we are going. So we would like to know how the mitochondrial network is changing with respect to drug treatments. And so what you have here on the top left, so that's essentially just the control mitochondrial network color-coded by diffusivity. So you have like bigger chunks that are moving slower, you have smaller chunks that are moving faster. And then what I have in the middle, that's essentially a transport simulation of what's happening. Like if you want to transport mitochondrial machinery within the network, can you actually reach everything, you know? Or is there like a piece of mitochondria that is just like somewhere that in the time course of your observation is never being reached? And so therefore material cannot really be exchanged with this. So this has classically been done with like these beautiful like uncaging of fluorescence or like injection of fluorophores. And then you see how like these fluorophores like distribute through the mitochondrial network. We can do this in silico on the networks that we have tracked essentially. And so then we can do like interesting experiments. We can say, well, in the wild type, it looks like we can actually reach the mitochondrial network quite well. And takes a little bit of time, but it's fine. But then if we essentially go ahead and we kill the most connected nodes in the network artificially, you know, we can measure the network and we say, okay, which are the nodes that are most connected? Let's just take them out. And how does the flux evolve in that case? So then you see that suddenly, you know, your transport, so transport efficiency is color coded. Red is like most effective, and like the lighter colors is less effective. So it takes much longer to distribute stuff through the network, which makes sense. And so then when you do this for the control, the effects are there, but they're not that much. Okay, so like this is wild type. This is you delete 5% random nodes. This is if you delete 5% most connected nodes. But when you do this with oligomycin is an ATP synthase inhibitor. So you essentially shut down the ability of the cell to make ATP. The cell reacts to this with a drastic disruption of the entire mitochondrial network. Okay, so before you have like this beautiful network, here everything is in fragments. Okay, so here this might actually look a little bit like these potatoes that you have in Google Images, but you know, not really. So everything is fragmented. So now we can really look at like how fast everything is distributed in the network, and it's really interesting. We see that everything is distributed much faster, mainly because these small particles, these small chunks of mitochondria, they're like diffusing much faster. They're fissioning and fusing much faster. And so suddenly, your reachability of the mitochondrial network goes through the roof. My hypothesis is a little bit that what we're looking at here is essentially an adaptation of the cell to ATP synthases being broken, and the cell trying to somehow scramble enough material and distribute it through the network to somehow make enough energy to actually still be alive so that we can look at it. Okay, so this might actually be the cellular adaptation, of this severe drug condition that we have here. And so but then here is also interesting. If you now go ahead and delete 5% of the most connected nodes, you can actually not reach most of the network anymore. So, it makes essentially the mitochondrial network much less resilient in that condition against random deletion of nodes. So yeah, those were the really interesting observations that we have with this. And so yeah, so that brings me to my final two slides. So here, the new work is essentially on the left-hand side again, the simulations and the modeling. We use the in silico simulations to do in silico mitochondrial simulations, and use them to validate our tracking tools. And so then the next step is obviously to do the same thing, use them with deep learning approaches. We work on that right now. Check out mitotnt.org. We essentially have all the code is on GitHub. This is a documentation of the code. This is a Jupyter notebook that essentially allows you to very easily access the Python library that is underlying all of this. And it's on bioRxiv since yesterday. And so the last thing remaining is then to really thank the team. So the leader of this project was Zach in the audience today, literally right in the middle. Zach's going to give tomorrow a live demo of MitoTNT. So if you're interested, come over. We're going to pull up the Jupyter notebook. If you have your own mitochondria data, we might actually be able to play with it. It would be fun actually to see how that plays out. Other people involved in the project were Parth over here. He did lots of he's a machine learning expert, did lots of the network analysis, and then essentially everybody was involved. And then yeah, these are the people who are giving us funding: Hardware, Keck, Research Corporation of Science, and Chen Zuckerberg. And then we have 2 job openings right now. So if you think this is cool, if you want to help, join us. We have a postdoc in mitochondrial cell biology, can work with stem cells, organoids, the microscope, and pick some of these low-hanging fruits. And then we also need a lab manager because now we have to essentially manage a couple more projects like this. So yeah, let us know if you know somebody who would like to live in in San Diego and join us. Let us know. Thank you very much.
Thank you, Bill. So, because Zach will be giving his tutorial tomorrow in the auditorium and the virtual people will be able to attend, and we're running late, let's just take one audience in-person question and one from Toby.
Thanks, Joe. It's really fantastic and exciting. I think we were in the same group, so we talked a little bit about what we do in the mitochondrial field. One thing I wanted to ask you a question, we can talk rest more because I have a lot more questions. Can you do it in vivo?
Yeah.
Excellent. So we have a mouse. We have a mouse model where we can tweak mitochondrial dysfunction or disturb mitochondrial function at will in any tissue.
Cool.
So we can follow up a little more.
Yes, absolutely. Yeah, so our lab is doing, we do everything in organoids because essentially we grow our own tissues because our learning curve is much quicker. But yeah, we will have a couple in vivo, not in vivo. We cannot really take a mouse and put it on a microscope. That's not possible, but it's going to be possible in a little bit. What we can do is essentially have organotypic slices. Like you take the live tissue, you slice it, put it in media. That works. Yeah.
Okay. Toby, do you have anybody lined up?
Well, there Robert, again, who's a prolific questioner, has provided one. Robert, I wonder if you wanted to chime in.
Yes, thank you very much for that talk. The question I had is how similar does the topology have to be? That is, how much of a constraint does topology need to be to result in accurate tracking? And for example, can this type of approach, the mito TNT that you showed, can it be used for other cellular compartments in which topology changes to a greater degree or greater extent over time?
Absolutely. That's a fantastic question. Thank you. The question is essentially, is this only applicable to mitochondria? Or can we actually look at all of these network structures in the cell? And my gut feeling is absolutely, we can absolutely, like we got a question immediately after we put this on bioRxiv about, can we use this on the actin network, like on the cytoskeleton? And I think my strong feeling is absolutely, because, you know, nothing really changes as quickly in the cell as the mitochondrial network, maybe with the exception of the endoplasmic reticulum. And so that means the topology constraint is going to be much, much stronger. And we have already seen that, like, if we switch off the remodeling of the mitochondrial network, we are doing much, much better in terms of accuracy of the tracking. So I think we should totally, like, just try it and go for it.
Okay, thank you.
Thanks, Joe. That was really, I'm just stunned into silence, I have to say. We're gonna move on to provocation 2. Just go back to your same table. If you're starving from that minuscule lunch and no breakfast, let me promise you that the poster session will have lots of cheese and crackers and lots of alcohol.
Before everyone leaves, I just want to point something out on Chi-Storm, because we will be discussing different questions in different groups, and you might have thoughts about the other questions that are posed on the other groups, and you might not be, thank you, and you might not be in that group, but you do want to share your thoughts. So before the day is out, you can add your thoughts to the other provocations. So if you click on this up here, it'll take you to each of the different questions that are being stewarded by different groups. And feel free to just comment and add questions or thoughts that you might have. So we'd love to hear your contributions as well. And we've had 2 phenomenal speakers, and a great project presentation. So think about that as you're in your small groups and thinking big with an unlimited budget. All right, see you all in a bit.
Jocelyn, are you going to allow me to
3:10.
disappear off?
Thank you.